The next talk will take us again to the human beings. And this is Maria Grazia Pizza, who comes from Siena, from GSK, and she's the director of bacterial vaccines. I think we should congratulate her because recently her vaccine for meningococci was approved and is now in use. And I'm sure that this will save a lot of children all over the world. And the next step, I think, is treating E. coli with vaccines. And the main question is, what happens when you vaccinate against something that's E. coli, like in our, very prevalent in our gut? And Maria Grazia will tell us what happens to the microbiome.
So, thank you, Eliora, and thank you all of you for this great opportunity to attend this meeting. I'm learning a lot. And now, as Eliora was saying, we shift in talking about vaccines and not antibiotics. So, and the effect on microbiome. Obviously, all the data we show you are related to mice because we are still at this stage with this project. But just to have an idea about the kind of questions we are trying to answer and which are the experiments we are planning to do. So let's start with the project. Okay, so just to give an overview of what happens to the different strains of E. coli. So as you know, there are different pathotypes, and the E. coli can be divided in pathogenic and commensal, even if this difference between pathogenic and commensal is not always easy. There are different pathotypes that are responsible for different disease, like we have the extraintestinal E. coli, which are mainly responsible for urinary tract infection. Sorry, I was looking for this one. Okay, so the extraintestinal E. coli, which are responsible for urinary tract infection, sepsis, neonatal meningitis, or intestinal E. coli, which are responsible for mainly diarrhea. And there are different pathotypes which are, as you can see, which colonize different sites into the human body. So with the idea to identify antigens which could be used as universal vaccines against E. coli, we started with this approach that the same that we have been applying to meningococcus B, that we have been calling reverse vaccinology, since it starts from the identification of antigens starting from the genome. And in the case of E. coli, since the project was more challenging, because as we said, we have pathogenic strains of E. coli and what is called non-pathogenic strains, we started to do genome analysis comparing strains giving different disease with non-pathogenic strains. So we did this kind of comparative genome analysis, and we also used proteomics as schematically shown here. This is a picture of a 2D gel in which we were looking for antigens which were exposed on the surface of the bacterium. So we have been doing this kind of analysis, trying to identify the antigens that could be good vaccine candidates. By this analysis, by using the bioinformatic tools, we were able to identify more than 200 antigens, and 43 of them, interestingly, were common to neonatal meningitis-associated E. coli as well as to uropathogenic E. coli. So we did this kind of analysis, trying to identify antigens which were conserved in more than one pathotype. All these antigens were expressed in E. coli, the proteins were purified, and the antigens were tested for protection in a sepsis model. So this is the summary table of all this, from all this screening, which were the antigens which were more protective in the animal model. And as you can see here, they have different numbers depending on their position on the chromosome. Some of them were homologous to known antigens, while other of them were not known in terms of function, and we had the protective efficacy, which was going from 80% for the main protective antigens to 30% for the least protective antigen. So on the basis of this analysis, we started to ask the question whether these antigens were present and conserved also in other pathotypes. And this is just to give you an idea about these are the different antigens and these are the different pathotypes in different colors. And as you can see, and the gray box means that the antigen is present in the different pathotype. White box means that the antigen is not present. And as you can see, there are antigens which are more present in more pathotypes, while there are other antigens which are present only in few pathotypes. So by this analysis, like in the case of our main antigen, it is absent in enterhemorrhagic E. coli, while is present in many of the other pathotypes. So on the basis of this analysis, our first conclusion is that probably a vaccine against E. coli should contain more than one antigen to be really able to cover the different pathotypes. Let's go to our main antigen, and I will give you the example of this antigen because it's the one for which we have started to do some analysis on the microbiome. So these antigens, which is 3526, is highly conserved in several E. coli pathotypes, but as you can see here, this antigen, the gene is also present in non-pathogenic E. coli. So when we went to look at the protection activity of this antigen, we saw that this antigen was in fact able to protect mice in different animal models, meaning in the intestinal colonization model, in the urinary tract infection model, as well as in the sepsis model. And this slide shows the comparison between the protection of mice immunized with SSLE, 3526, and saline. So as you can see that there is different degree of protection in the different animal models. And we were also able to show that these antigens was able to have passive protection, in a passive protection experiment, was able also to induce protection in the chicken model of sepsis. And this experiment was done in collaboration with Eliora Ron here at Tel Aviv University, in which you can see that when the chick were given, the antibodies were given to chick, we had a good protection. Compared to the premium sera. So we have been shown that this antigen is, as I said, able to induce protection in the different animal model by active and passive immunization. But what this antigen do, just a few slides to give you more detail on the possible function of this antigen. So this is a lipoprotein, it has a lipidation motif, has a lot of other activities like zinc metalloproteases activity, and a proline-rich region, an RGD domain which seem to mediate adhesion to epithelial cells. But interestingly, what happens is that this antigen is a lipoprotein but is also secreted. And what is interesting is that this antigen is expressed by non-pathogenic E. coli, by commensal. As you can see here, this is a Western blot showing the expression of this antigen, while it is non-secreted in these non-pathogenic strains. So this means that this antigen, for a known reason, is expressed by pathogenic and non-pathogenic, but is only secreted by pathogenic E. coli strains. So on the basis of these results, we can say that okay, these antigens that, as I said, was before there was a number, but now we know the function, and this is why this has been named SSLE for secreted and surface exposed antigens. This antigen provides protection in different animal models, is able to degrade mucins. We identify the function. and is able to induce antibodies which prevents in vitro mucin degradation and is able to translocate through to prevent translocation the antibodies through the mucin layers. So the question is, but since this antigen is present also in non-pathogenic strain, so can immunization with these antigens influence the gut microbiota? So, as you know, there is a lot of discussion. We have been discussing before the effect of antibiotics on microbiota, but obviously there is a lot of ongoing on the relation between vaccines and microbiome. Because as we know, that as we have been hearing before, the intestinal microbiome has also an important role in shaping mucosal and systemic immunity. And we have seen also that the microbiome has an important role also in many inflammatory bowel disease. And we also know that microbiome may affect vaccine response. In fact, there are many studies in which they've been trying to understand whether differences between responders and non-responders is due also to the different microbiome composition. And the majority of the studies has been done mainly on rotavirus vaccines, where they've been looking whether in the different population there is a different immunoresponse also because of the microbiome composition. So although there is no a final answer to this, because as you can imagine, it's quite complex to find a relation, but what is important to note is that the microbiome composition before immunization in the different population is in fact different, and this can induce obviously also differences in the immunoresponse. So obviously the question here is how microbiome influence vaccination, but the question we have is vaccines may also affect the microbiome. And working for a company that wants to make vaccines, obviously this is an important question. Are we going to impact the microbiome if we induce antibodies against an antigen which is present also in the commensals? So, considering the presence of this antigen in many coli commensal, the question has been, is there an effect of immunization on the resident gut microbiome? And this is the immunization protocol, so mice have been immunized 4 times by using different immunization routes. Actually, we have been using the intramuscular immunization, using an oil in water adjuvant, or intranasal immunization to induce a mucosal immunoresponse, but also a combination of intranasal immunization at the first and then intramuscular immunization for the following 2 immunizations. And we have been looking for local and systemic immunoresponse, and also looking for intestinal lavage and feces to look for immunoresponse, but also for microbiome analysis. So the data in terms of immunoresponse are schematically shown in these slides, where we have been measuring the total IgG response, so the total antibody response in the serum, the different subtypes, so the ratio between the different subclasses of antibodies, but most importantly also to the IgA response, the mucosal immunoresponse, both in the intestinal washes and in the fecal pellet. And by this analysis, we have demonstrated that the mixed immunization, meaning the first immunization mucosal, intranasal, and the other 2 immunization systemically intramuscular, gave the higher immunoresponse both in terms of IgG and IgA. So because of this data, we decided to proceed for the microbiota analysis, and we collect samples at day 0 and day 71, meaning before immunization, at the end of the immunization cycle, and we did Illumina sequencing looking for the 16S ribosomal gene, and more than 65,000 sequences were recovered from these samples. So these are the analysis of the microbiota composition from both cecum and feces of mice immunized with the we decided to look for the immunization protocol for which we had the highest titer of antibodies in terms of IgG and IgA. And as you can see here, we don't see too much difference comparing the pre-immunization and post-immunization in mice which were immunized with the saline, so without the antigens, or with the antigen. And this is also confirmed by the Shannon Diversity Index, where you can see that there is not much difference between these two groups, PBS or SSLE immunized. If we look at the microbiota composition, so we can see that there is not, even if there is a different percentage, I would say, between some of the families, but we can see that the percentage overall remain almost identical. And this is true for, less true for the fecal pellet, but this is quite undistinguishable in the cecal content, as you can see here. There is a difference, as you can see, between pre-immunization and post-immunization, particularly in the Firmicutes family, but this could be due to the stabulation, the animal house, because these mice were transferred from the vendor to our animal house, and as you can see, there has been a reduction on this firmicutes. So it seems to be linked probably to what the mice eat and the condition they have in the animal house. So in conclusion, we can say that there were no significant changes in the overall richness in the microbiota between mice immunized with PBS or SSLE. The same families in general, where general bacteria were recovered between the two groups, pre and post immunization. As we have said, there is a relative abundance of firmicutes that seems to be different pre versus post, but this can be due to the transfer of mice to a different facility. There were no differences between lactobacilli and lactospiraceae in the cecum, and we can conclude that mucosal immunization, at least in this protocol with SSLE, does not perturb the gut microbiome. And just to finish, I want to acknowledge all the people that have been obviously involved in this project since the beginning, starting from the identification of the antigens. Ilham, which was a PhD in our lab and which has done all the work on the microbiome analysis. And this work has been only possible because of a lot of collaborators we have in the different laboratories with different expertise. And thank you for the attention.
Thank you, Maria Grazia. So now we can safely take the vaccine when it comes out to the market? Okay. Yes, George. Microphone.
I just talk louder. So,
Yeah,
okay, thank you for a very nice talk, Maria Grazia. Did you just look in normal animals or anything without any vaccination or anything to see if there was IgA or IgG against this antigen?
Yes, we have done. We have done it in the past. There are no antibodies. We were quite surprised at the beginning, but we never see. And generally, for our analysis, we always use mice that have been in the same animal house for the same time, and we use them as control. SSLE, there are antibodies in humans.
Okay, and if you induce colitis or do something to make it dysbiosis, Do you see antibodies appearing there?
We have not tried. This is an interesting experiment to do. Yeah.
It is obviously very encouraging that this is both protective and non-disruptive to the microbiome. What is the, just elaborate on what is the target market for this vaccine? What human populations will or should be vaccinated once it, clears all safety issues?
So as you can see, as you can imagine, this is a never-end discussion. So originally we were targeting extraintestinal infection, and this is why also we started with those genomes. And the idea for extraintestinal infection was mainly urinary tract infection. So the idea was to target that population, meaning adolescents or people with recurrent infection. But then we realized that these antigens are conserved also in other pathotypes, and for this reason we are thinking to something more broad, but will be difficult because you have to do efficacy trial anyway against the different pathotypes you are targeting. So for now we are mainly looking for as enterotoxigenic or UTI infection.
I'll ask a question too. You think that the results would be different if this protein was secreted by the non-pathogenic ones?
This is a great question, Eliora, because we are still, for us, it's still difficult to understand why a protein should be surface exposed and secreted, because it's quite strange that you have the two signals. And it's even more strange that commensal don't do that. So this means that probably this protein has completely different functions depending by the pathogenicity of the strain. Yeah.
Thank you. If there are no more questions, well.